Good afternoon, everybody. So it's great to be back here. Thanks, OIS organizers, for inviting me here to present again. The last time I presented was in New York in 2022. And now I'm back here to present our first time ever clinical results. So our concept is unique. What we do is we design small molecules that specifically activate our resident, hence endogenous stem and progenitor cells to get activated, and then they migrate to the site of injury, to the damage. They mature into the functional cells and restore, hence, the structure and function. And our concept is applicable to all kinds of organs and tissues across our body, but we obviously focus first on the eye, but have also projects on lung cell regeneration and blood cell regeneration. It's an innovative approach because it really focuses on the outcome of this regeneration and function of the newly produced cells. Our compounds are AI designed and they are novel. So we have composition of matter patent for all of our molecules. We have a broad therapeutic potential, as I mentioned before, because it's a general applicable approach that we pursue. Some of the advantages are we are gene and target pathway agnostic. It's very easy to administer. It's a small molecule, easy to manufacturing with low cost. And with that, out of that platform, we have created EA2353. The profile of this molecule, it's a first-in-class small molecule endogenous regenerative medicine. It activates the dormant retinal stem and progenitor cells in the ciliary marginal zone, and it's for treatment of RP, or in fact, any inherited retinal degenerative disease, regardless of the underlying mutation. So the first in-human study demonstrated that it is safe, and we have preliminary efficacy that I will guide you through. So firstly, just to recapitulate quickly, repeat what the preclinical findings were. So we were able, in blinded mice, to regenerate structure, but also functions. So you see, we were able to regenerate rods and cones in blinded mice that also led to very nice visual function regeneration. Quickly, all of this has been published by now. So at the bottom, you see the publication where you can read up on that. So our first in-human clinical trial was a dose escalation study, 3 dose escalation groups, and then an expansion cohort. Preliminary outcome was, of course, safety and tolerability, and I'm very happy to report that we have no cases of intraocular inflammation related to the study intervention, and we have no relevant ocular findings or even systemic. We have no systemic exposure. Then we had a number of efficacy endpoints that we were looking at as well. So I guide you through some of them. So we have seen in some patients, we have seen structural improvement in ONL as well as ELM, and you see an example here measured by OCT. We have observed visual acuity improvement in BCVA, and we have also observed visual improvement in low luminance visual acuity, which obviously is very important and relevant for retinitis pigmentosa patients. What is very exciting also is what we found with microperimetry measurements. So on the top, you see the treated eye, and in the bottom, you see the control eye. What you can see is obviously that we were able to get something from nothing, so you have a true regeneration effect here as measured by microperimetry. So I've shown you individual examples, but actually I'm very happy to say that we have seen this improvement through all cohorts and relevant through all different kind of measurements. And of course, I'm happy to tell you more about it. Please come and see me afterwards. What is also important is we have a patient-reported outcome measure, and we have what for me was really relevant and for all of us is that we have very positive feedback from the patients that reported an improvement in their eyesight. So in summary, the compound can be administered safely, no ocular inflammation, no relevant ocular or systemic safety issues. We have seen structural improvements, as shown by increases in subfoveal ONL thickness and ELM width, and seen in patients from all cohorts that we have tested. Improved central retinal sensitivity was also observed in microperimetry, again from all cohorts, as well as visual function improvement. So hence, we are now well positioned for the next study, which will be a phase 2b/3 pivotal trial that we are currently about to set up. So this is also the time for me to thank all the patients and investigators that participated in this first clinical study. But this is not all. So we have a second program that is targeting dry AMD, GA, and it's another compound. It's EA2351 because we target the different progenitor cell source. We target different cells, we target different pathways. This molecule is also novel, and we have demonstrated RPE regeneration in sodium iodide rats that you can nicely see at the bottom, where the RPE really is growing back from the outside. That actually led also to visual function improvement. So if you want to learn more also about these preclinical studies, please come and see us at the poster that we will be presenting on both of the projects on Wednesday. Thank you very much.